I will now introduce Dr. Serena Chierici graduated in chemistry at the University of Pavia in 2014. She's a researcher at the Experimental Station for the Food Preservation Industry Packaging Department in Italy since 2015. She deals with European projects focusing on the valorization of food processing waste and particularly of tomato byproducts. She's also involved in the study and evaluation of the packaging materials in terms of shelf life and hygienic sanitary compliance. She will share with us the presentation Recovery of Added Value Molecules from Agro-wastes to Apply as Coating for the Food Packaging. Please, Serena, the floor is yours.
Thank you very much. Good morning, everyone, and thank you, Mara, for introducing me and for giving to SSICA this opportunity to share this the experience of SSICA in this EcoFanco project. So in this brief presentation, I will describe the activity carried out here in SSICA based on the recovery of added values molecules from agro-waste to be used as starting material to formulate coatings for the food packaging application. But before start, just a quick overview about the background of SSICA and the expertise then applied on the EcoFanco project. So SSICA was and is still involved in several European projects in which the extraction of added values molecules is performed from agro-waste. So the valorization of the agro-waste was carried out. In particular, the agro-waste selected from SSICA are legumes and tomatoes. So the expertise and the experience started from the laboratory scale, of course, but in these years can achieve also the pilot and semi-industrial scale as already anticipated by Patrizia. So this know-how was exploited also in the EcoFanco project. In this project, the target substance is the cutin, which is a polyester mainly composed of long-chain fatty acids. This substance is found in the cuticle of fruits and vegetables and has several functions. One of them is protecting one from the external agents and mainly from the water loss of the fruits and vegetables. So it's a preserving substance for these vegetables. So based on the cutin abundance in the cuticles and on the fatty acids profile, which is different among the different type of cuticles and vegetables, and also considering the availability of the vegetable source, so in order to give an valorization of these byproducts, 3 different agro-wastes were selected for the extraction of the cutin in the EcoFanco projects. So the 3 byproducts were tomato, watermelon, and apple peels. Concerning the tomato peels, SSICA already has experience in the extraction of these feedstock, but to achieve an optimization of the protocol, several tests and trials were carried out during the years also, changing the time and temperature conditions. So at the end, SSICA set up protocol avoiding the organic solvent use and applying few and easy steps. As you can see, in fact, in the flow chart, the peels and seeds are separated, then the peels undergo to a thermal treatment, which is the most important step in which the cutin is extracted, in fact. Then the liquid and solid are separated, and the liquid phase is acidified in order to favor the precipitation of the cutin, and its recovery is done. The extraction of this molecule and also the setup of the condition always focused on the scale-up perspective. So time, temperature, energy and resources consumptions were also the most important thing to remember in order to have a scale-up possibility. In addition, also an additional valorization could be done by using the waste generating during the extraction process, allocating them to the biogas production and consequently to energy production. After all the trials, yields of extraction achieved the 40% in weight, which is a very important and significant result. So the success of the extraction was monitored by the FTIR analysis. In this case, the spectrum, the identification of the characteristic peaks of the cutin were done. So the main peak is the one of the stretching of the C double bond O, which is more or less at 1,700 centimeter, and also the other peaks as indicated in the spectrum. Consequently, the characterization required also the identification of the fatty acid profile of the cutin. So a GCMS analysis was necessary. In the case of the tomato cutin, the main component of the bulk is the 1016 hydroxyhexadecanoic acids, which is the major fatty acid in the substance, as indicated in the chromatogram. The other peaks are identified as minor fatty acids. Considering the watermelon peels and apple peels, SSICA extracted the cutin at laboratory scale. These are new sources tested. Also, in this case, the protocol tried was the one set up for the tomato cutin, but different some changes should be applied to adequate the feedstock, the source, to the extraction. So time, temperature conditions were evaluated, different conditions were evaluated, but also the sodium hydroxide concentration, which is the extracting solution, was tested in order to achieve the best results possible. As before, the sustainability of the protocol was taken into consideration in a perspective of a possible scale-up. After the testing of these sources, the yields of extraction achieved a 20% in weight of the apple peels, which is a very positive result, while for the watermelon peels only a 2% can be achieved. So in this case, further research are required in order to improve this extraction for the watermelon peels. Also for these cutins, the first evaluation for the extraction was performed by the FTIR analysis. Also in this case, the identification of the characteristic peak of the cutin were carried out. Concerning the apple cutin, positive results are achieved because no significant impurities were detected in the spectrum, while concerning the watermelon cutin, a significant influence of the impurities were detected, as highlighted by the red circles. So again, concerning the watermelon cutin, further research are necessary to improve this result. Secondly, the fatty acids composition of the samples of apple and watermelon cutins were carried out by GCMS analysis. In this case, some differences for both type of cutins were highlighted, comparing them to the results available in the literature, which are, I have to remark, very, very scarce. So not big, not lots of papers are available for this type of source for the cutin extraction. So in this case, we found that there is differences between the proportion among the fatty acids composing the watermelon and apple cutin.
Excuse me, Serena, you have 5 minutes left.
Okay, thank you. So another thing to be added is that the as you can see in the table, watermelon and apple cutin have a more complex composition compared to the tomato cutin. In fact, we have no predominant fatty acids in the composition, but we have several fatty acids with similar abundance. So also in this case, it's not so easy to achieve a very positive result as those for the tomato one. Another thing that can be added is that with SSICA protocol, we avoid organic solvent extraction in a perspective of a sustainable process and protocol. While in the literature, the protocol available used always solvent and also long time duration extraction, composed of many steps and sometimes very complicated steps. So why we want to use the cutin in this project? So as anticipated, cutin has hydrophobic properties itself. So the goal is to transfer this water repellence property to substrates as bioplastic and cellulose, which do not have enough barrier to be used as food packaging. How we can achieve this ambitious objective? Well, combining the cutin with other ingredients and components in order to formulate and give the cutin an adequate, suitable consistency to be applied as a thin layer on the selected substrates, and by using the traditional application technique already available in the specific industries. So concluding, cutin was successfully extracted from tomato peels, both at laboratory and pilot scale, avoiding the use of organic solvent, which is a very important result. Also, apple and watermelon cutin were extracted, applying the same method of the tomato cutin with some changes. But in this case, some differences were observed in the fatty acids profile, comparing them with the literature. So in this case, further research are required to improve the results and also to evaluate the differences observed. And at last, cutin, being a natural hydrophobic substance, it is a very interesting substance to be applied to confer water repellence property to bioplastic and cellulosic substrates. So thank you for your attention.
Thank you, Serena, for your presentation, and thank you for presenting us the details on the processes that SSICA is implementing for the cutin extraction.